Bismillah ar-Rahman ar-Rahim. Female reproductive cycle. The cycle starts at puberty. It continues throughout the reproductive years. This cycle involves activities of first the hypothalamus, ovaries, uterus, uterine tubes, vagina, mammary glands. Considering the hypothalamus, it acts on cells of the anterior lobe of the pituitary gland. In turn, it secretes gonadotropins, including the follicular stimulating hormone and luteinizing hormone. These stimulate and control the cyclic changes in the ovary. These are the following functions which are performed by the female reproductive system. First, formation of eggs, reception of the sperms during copulation, providing a conducive environment for fertilization, providing shelter and nourishment to the growing embryo. First week of development. It includes ovulation to implantation. The ovulation, it means that the egg is released into the peritoneal cavity, traveling down the fallopian tube in which the fertilization occurs. At conception, in the fallopian tube, maternal and paternal genetic materials join together, forming the new human life, which is named the zygote, containing the diploid number of chromosomes, half from the mother and half from the father. Immediately, cell division starts with it traveling down the tube and into the uterus. The ovarian cycle has two distinct phases. The follicular phase starts from day 1 to 14, the luteal phase starting at day 14 to the 28th day. It begins with the formation of the corpus luteum, ending with either pregnancy or luteolysis. Both follicular stimulating hormone and luteinizing hormone, they produce cyclic changes in the ovaries. They cause development of the follicles inside the ovary, then ovulation by expelling the ovum outside the ovary, then formation of corpus luteum. Ovulation, it occurs around mid-cycle, about 14 days, in a 20-day menstrual cycle. The ovarian follicle undergoes sudden growth, spurt or burst under the influence of the previously mentioned hormones, the follicular stimulating hormone and the luteinizing hormone. The expulsion of the oocyte results from raised intrafollicular pressure. This oocyte will be surrounded by zona pellucida, which is a layer of glycoprotein material, and layers of follicular cells. These cells are known as the corona radiata. The zona pellucida is composed of glycoproteins, as I mentioned. You can see in this slide, this is the ovary. Here are the developing follicles. Usually the ovary forms about 15 to 20 follicles. Only one of these follicles will continue to grow and reaches maturation. The rest are degenerated. So this is a primary, secondary, and tertiary follicle. Here is the oocyte inside the follicle, surrounded by the follicular cells. Once the expelling of the oocyte starts, these follicular cells detach from the lining of the follicle. They surround the oocyte. These are named the corona radiata cells. Here you see the oocyte is surrounded by the zona pellucida. Then the wall of the follicle will show foldings. It will change into the corpus luteum. The corpus luteum acts at this stage as endocrine gland, secreting sex hormones like progesterone and estrogen. Later on, it is named the corpus luteum, will be degenerated, changed into fibrotic tissue, named the corpus albicans. So, starting from the mature follicle, named the Graafian follicle, changing to the corpus luteum. Lastly, it will be changed into fibrous tissue, which is corpus albicans. As I mentioned, the corpus luteum, after ovulation, its walls of the ovarian follicle collapse and thrown into folds, which is named the corpus luteum. It serves as temporary endocrine gland, releasing progesterone and estrogen. Before any process of fertilization, the sperm undergoes a process of capacitation in response to conditions in the female reproductive system or tract. This includes increases in the motility and destabilization of the cell membrane that allows the head of the sperm to penetrate the egg. So the sperm have to pass into this stage so it will be able to fertilize the egg. So capacitation is defined as the period of conditioning in the female reproductive tract. Much of this conditioning during capacitation occurs in the uterine tube and involves epithelial interactions between the sperm and the mucosal surface of the tube. So only the capacitated sperm can pass through the coronal cells surrounding the oocyte and undergo acrosome reaction. In turn, the acrosome reaction, it occurs after binding of the sperm to the zona pellucida. It is induced by zona proteins. This reaction culminates in the release of the enzymes needed by the sperm to penetrate the zona pellucida, including acrosine and trypsin-like substances. The mammalian female reproductive tract interacts with the sperm in various ways in order to facilitate the sperm migration to the egg. In order to, first, it will keep the sperm alive during the time between mating and ovulation. Second, it will select the fittest sperm for fertilization. You know that hundreds, millions of sperm, they enter the genital tract of the female. Only about 200 to 300 will stay in the area of fertilization. Then only the fittest sperm will be selected to fertilize the ovum. The two main types of interactions are physical and molecular. The physical interaction includes swimming responses of the sperm to the microarchitectures of the walls. It means the walls of the uterus or uterine tube. When the sperm encounters wall, they have a strong tendency to remain swimming along them, so it can pass freely and move freely through the uterine tube until it reaches the ovum. The female tract seems to use these tendencies of a sperm to guide them to the site of fertilization. The sperm at this stage moves freely inside the tube until it reaches the ovum. Now, fertilization passes in three phases. Phase 1 includes penetration of the corona radiata. As I mentioned, about 200 to 300 millions of sperms are deposited in the genital tract of the mother. Only about 300 to 500 they reach fertilization site. Only the fittest, as I mentioned before, the fittest sperm will be able to fertilize the oocyte or the egg. Phase 2 includes penetration of the zona pellucida. Here, the zona is, as I mentioned, the glycoprotein shell surrounds the egg. It facilitates and maintains the sperm binding, inducing the acrosome reaction. By releasing acrosomal enzyme, acrosine and trypsin-like enzymes, these enzymes will allow the sperm to penetrate the zona. thereby coming in contact with the plasma membrane of the oocyte. Then again, there is release of lysosomal enzymes. These enzymes secreted by the cortical granules lining the plasma membrane of the oocyte. The function of these enzymes, they alter the properties of the zona pellucida. This is the zona reaction. Then phase 3 of fertilization involves a fusion of the oocyte and sperm cell membranes. After adhesion of the sperm, the plasma membranes of the sperm and egg fuse. Actual fusion is accomplished between the oocyte membrane and the membrane that covers the posterior region of the sperm head. Here again, this is the ovary. As I mentioned, this is the primordia of the follicle. It will grow, only one will grow and reaches maturation. And it is named the Graafian follicle. Later on, after expanding of the oocyte with the cells of the follicle or the follicular cells, which are the corona radiata, and it's also surrounded by the zona pellucida, the folding of the walls of the uterus the follicle will occur at this stage and will change to the structure named the corpus luteum. Corpus luteum, and after it finishes its function as endocrine gland, it will change into the corpus albicans and then degenerates. The luteinizing hormone surge from pituitary gland. It causes changes in tissues and within the follicle. There will be swelling within the follicle due to the increased hyaluronine and matrix metalloproteinases. They degrade, surrounding and causing the rupture of the follicle. These are responsible for the expanding of the intrafollicular pressure that will aid in the rupture of this follicle, so will release the oocyte. Corona radiata provides bulk to facilitate capture of the egg, as you can see here, to be captured by the fimbria. Here again, this is the oocyte surrounded by this is the zona pellucida, and here is the corona radiata cells. At around the midpoint of the menstrual cycle, about 14th day, this egg is ovulated and swept into the oviduct. Here, this is the ovary. Here, the fimbria of the uterine tube will capture this released ovum and will take its pathway through the uterine tube until it reaches the endometrium of the uterus, where it will be implanted. The fertilization, here you can see the fertilization starts here. It occurs in the ampulla, one of the segments of the uterine tube. Uterine tube is divided into 3 segments. The ampulla is one of the segments of the uterine tube at which the fertilization occurs within 24 hours of ovulation. Then, once this occurs, this will start the cleavage process, in which the division of cells will start, where the series of cleavage, which is the process of dividing of the cells, differentiation events occur and result in the formation of the morula, or the last stage, then the blastocyst. Here you can see, here after copulation or fusion of the both gametes from the paternal and maternal source, here they start immediately, division occurs, 2 cells, then 4, 8, until reach about 16 cells. This structure is named the morula. This morula is a solid ball of cells. Each one of these cells is named the blastomere, surrounded by this blue color circle. It is the zona pellucida. Later on, here, changes occur in these cells. They start to differentiate, forming the blastocysts. Then, here, implantations occurred at this area. You can see the blastocyst is implanted in the endometrium, or the inner lining of the uterus. Here again, the fertilization is a multi-step process whereby multiple sperm bind to the corona radiata, but only one or a single sperm usually fertilizes the egg, including to the acrosome reaction and the Zona reaction. Here the sperm reaching the area where the corona start penetrating the corona radiata, then it binds to the to the zona pellucida, penetrating the zona pellucida until fusion occurs between two plasma membranes of the oocyte and the sperm. Again, this picture shows the phases of fertilization. These are the cortical granules where they release the lysosomal enzymes, so allowing the sperm to penetrate the zona pellucida. Here, half number of chromosomes, and both of them will unite together, forming the diploid number of chromosomes. Once this occurs, the fertilized egg will start, or the zygote start to divide. Here, the fertilized egg, day 1. This is scan electron microscope. Then 2-cell stage. One cell gave 2 cells. The cells start to decrease in size. They increase in number but decrease in size. Each one of these cells are named the blastomere. This is the 4-cell stage. Also, these blastomeres decrease in size but increase in number, still surrounded by the zona pellucida. The 8-cell stage, or the third cleavage, is named the third cleavage. Blastomeres are still equivalent. Then embryo undergoes compaction after 8-cell stage. This compaction is caused by increased cell-to-cell adhesion. The cells that are forced to the outside of the morula destined to become the trophoblast or the outer cell mass, forming the blastocyst. The inner cell mass will give the embryo. As you can see, this is the morula, and it's the stage of compaction starting here by the cell adhesion, and this is the zona pellucida. Here again, they're starting to cells start to differentiate by inserting this fluid inside this structure. This fluid will push these cells, some of them, to one poles of this structure. These cells are polarized. They form the inner cell mass. They will give the embryo. The outer cell mass, this single cellular of latent cells named the trophoblast. This stage gives the blastocyst, which is composed of, as I mentioned, 2 cell masses. The outer cell mass, which is the trophoblast. The inner cell mass is the embryoblast. Still, the zona pellucida is present. This structure is named the blastocyst. So blastocyst is divided into 2 cell layers, the outer and the inner, surrounded by the zona pellucida. Here, the fluid is pumped into this embryo. At this stage, this blastocyst is named also the blastocele, since it contains the fluid. Here again, this is another figure, shows the trophoblast and the embryoblast, the outer cell mass and the inner cell mass. Here, this is the endometrium of the uterus. The blastocyst is trying to reach this area and will be implanted here. Zygote leads to the formation of the morula, and then the morula gives blastocyst. This is the normal development. The blastocyst, I mentioned, it gives trophoblast and the embryoblast. The embryoblast, at this age, it means the beginning of the 2nd week. It will divide into 2 distinct epithelial layers just before implantation. The first layer is the epiblast. The second layer is the hypoblast. It's named the week of 2, for this reason, that the embryoblast give hypoblast and epiblast. The trophoblast, which is the outer cell mass, it divides into cytotrophoblast and sensatio-trophoblast. Also, here the cavities are divided into amniotic cavity and the primitive yolk sac. That's why it's named, the second week of development is named the week of twos. Here you can see this is the epiblast and this is the hypoblast, both forming the embryoblast. Here is surrounded by the trophoblast, which is divided in turn into cytotrophoblast and sensatio-trophoblast. The two cavities here, the amniotic cavity overlying the epiblast. Here is the site, will be site of the yolk sac below the hypoblast. So in general, this stage of development of the embryo, it's named the bilaminar disc at this stage. It refers to that this embryo is developed into two layers, the epiblast and the hypoblast. That's why it's named the bilaminar disc or the bilaminar germ disc. These two layers are sandwiched between two cavities. This is the amniotic cavity and later on the yolk sac. This second week starts from day 7 and 14 till 14th day of where the implantation occurs. The implanted embryo becomes more deeply embedded in the endometrium of the uterus. Then, further development of the trophoblast. As I mentioned before, the trophoblast, it represents the outer cell layer of the embryo of the blastocyst, which divided into cytotrophoblast and sensatio-trophoblast. This will further develop into the placenta. It will give the placenta. Development of the bilaminar embryo, which is composed of the epiblast, and epiblast, it's overlaid by the amniotic cavity, underlaid by the yolk sac. Here you can see this is the embryo, the embryonic disc, this one. This is the wall, to represent the wall of the uterus, the endometrium. And here the blastocyst is embedded or implanted in the wall of the uterus. Here the inner cell mass divided into epiblast and hypoblast. The trophoblast, I told you that's divided to cytotrophoblast, then sensatio-trophoblast. The sensatio-trophoblast is very important. It's composed of invasive fused cells derived from the cytotrophoblast, which is the other division of the trophoblast. The trophoblast composed of cytotrophoblast and sensatio. So the second division is the sensatio-trophoblast, which breaks maternal capillaries. Trophoblastic lacuna fill with the maternal blood. The embryo will join the trophoblastic shell by narrow connecting stalk will change or develop into umbilical cord attaching the placenta to the embryo. Here you can see this is the bilaminar germ disc. The blue color represents the epiblast. This color represents the hypoblast. These both forming the bilaminar germ disc. This is the amniotic cavity overlying the epiblast, and here this is the site of the yolk sac underlying the epiblast. These two layers represent or originated from the trophoblast. This is the cytotrophoblast and this is the sensatio-trophoblast. They will further develop inside the endometrium of the uterus. Here, some blood vessels will develop here, and it comes from the mother circulation in order to provide nutrition to the developing embryo. About 9 days of human, this is human blastocyst. This is again the epiblast, the hypoblast. These two structures represent the bilaminar germ disc. And this is the cytotrophoblast and sensatiotrophoblast. You can see here lacunae are developed in this layer. These are empty lacunae. They will receive the blood coming from the mother circulation. Since this developing embryo needs nutrition to grow. Here again, about 12 days of age, the trophoblastic lacunae at the embryonic pole are now open in connection with the maternal sinusoids in the endometrium stroma, starting to be filled with blood, as you can see. Further development shows the human blastocyst, trophoblastic lacuna, and the uteroplacental circulation. Here you can see these developing lacuna are completely filled with the blood to provide nutrition to this embryo. This represents the embryo. As you can see, here it is named the bilaminar germ disk at this stage, composed of two layers, epiblast and the hypoblast. This is the amniotic cavity and the yolk sac. This structure is named the connecting stalk. It will change into umbilical cord, connecting this developing embryo with the placenta. Then later on, as you can see, this is the bilaminar germ disk. This is the epiblast, and this is the yellow color represent the hypoblast. At this stage, the developing embryo is known as the bilaminar germ disk since it's composed of two layers only. Here, this is a localized area. At this bilaminar disc, it will form the precorder plate. The cells become more columnar at this area only. It is named the precorder plate. Here, when the bilaminar germ disk reaches this stage, it will occurs at the end, about the end of the 14th day. We are going to start the third week of development, where the gastrulation starts. At the end of the second week, trophoblast has had a period of growth greater than the embryoblast. The two-layered bilaminar disc is formed and will give rise to other tissues and structures. This is the embryonic disc. This structure shows a histological section taken from an embryo. Some clinical correlates only. I'm going to talk about the implantation site usually occurs normally or usually at the posterior or the anterior wall of the body of uterus, whereas the abnormal implantation site, or its name is named the ectopic pregnancy, may be seen at the abdominal cavity, mesenteries, or ampullar region of the uterine tube, which is one of the segments of the uterine tube. Now, thank you for your listening.